Hello everyone, my name is Selemani Sango from Dar es Salaam, Tanzania. I'm here to represent the PowerPoint presentation of the debate as a Group O that we made the debate against the Group P about the topic, should livestock farming in Africa be genetically monitored for AMR to prevent zoonotic transmission to humans? I, Selemani Sango from Tanzania, and my fellow colleague Bashiri Meriko from Malawi. We have the introduction. Livestock farming is vital for food security and economy in Africa, according to Ida Makoro 2023. Africa is one of the continents that heavily rely on livestock keeping and farming, according to FAO records of 2020. It contributes approximately 35% of the agricultural gross domestic product (GDP) in sub-Saharan Africa and supports the livelihood of roughly 70% of the rural population, according to EDRO 2023. Livestock farming is a major source of zoonotic diseases transmission, pathogens that can be spread from animals to humans. This report of Epsona et al. Of 2020 has reported these issues of zoonotic transmissions. 75% of all emerging diseases and 61% of all infectious diseases are zoonotic in nature, according to Dr. Hermani 2023 and 2024 said et al. Rising AMR due to antibiotic advice exacerbates zoonotic diseases, especially due to treatment failure. Opening statement. WHO describes antimicrobial resistance as one of the three most critical public health threats of 21st century. AMR-driven zoonosis rising at the alarming rate, straining under-resourced health systems in Africa, according to MBN Martins of 2025. By the year 2050, AMR infections to cause 10 million human deaths annually, mostly in Africa, if the present-day developments of antimicrobial resistance persists, according to Meriko et al. 2025. Genetic monitoring stands as a proactive shield that could be crucial against the next zoonotic pandemic. Our stand is advocating for genetic monitoring of AMR in livestock farming as one way of reducing zoonotic transmissions to humans. We have some arguments that we prepared for the debate. The first argument is early detections of pathogens. Using tools like WGS and metagenetics to identify and track resistant pathogens in animals, their waste and environment real-time Kegness et al. 2025. Allow the early detection of emerging resistant threats, silent reservoirs before they become widespread in human population. Case study we have in Vietnam. FMDV clusters circulating were found in samples and clinical healthy animals. Slaughterhouses acted as a transmission window or early warning points. They captured genetic material for calculating viruses even before outbreaks were officially detected. For some clusters, the virus detected in slaughterhouse samples appeared 4 to 6 months before the same cluster was identified in outbreak sequences. We have the argument to early detection of AMR genes, provided critical insights to the environment, reservoirs and AMR, like water bodies, hospital effluents, and livestock farming areas that harbor a diverse array of resistance genes, some of which have potential to be transferred to human and animal pathogens, enabling rapid quarantines and antibiotic stewardship. We have the case study in Canada. Metagenomic studies showed that untreated wastewater contains diverse resistance genes, including those conferring resistance to the last resort antibiotics like costly. These findings have informed policies to improve the wastewater treatment process, reducing environmental antimicrobial resistance reservoirs, according to Genetically 2025. Adapting the same in livestock farming can help to reduce AMR-related infections in the human population through reduced consumption of livestock products with drug resistance genes of pathogens. Argument 3. One Health Genomic-driven AMR modeling and surveillance. AMR prediction models which have blended genomic data with epidemiological, clinical, and environmental information to the forecast and emergence of spread-resistant pathogens. This model helps public health and authorities to anticipate AMR trends and take preemptive actions, such as optimizing antibiotic stewardship and updating treatment guidelines based on predicted resistant prevalence, according to Chigzi 2025. Case study we have, the genomic data has been used to predict the spread of carbapenem-resistant antibacterial diseases in health setting, allowing the hospital to implement targeted interventions as enhanced screening and isolation procedures for high-risk patients. In agricultural settings, AMR prediction model has been traced to assess the risk of resistance transmission from livestock to humans, informing the policies of antibiotic use in food production according to Penge of 2022. Argument 4, genetically resistant animals. Genetic monitoring can help to identify the animal species which are resistant to some strains of pathogens that are commonly implicated in zoonosis. Through selective breeding, we can lower antibiotic use and resistance by having only resistant animal species to certain pathogens. This can reduce the risk of zoonosis at large due to specification of the genetic who can now detect the change in genetics and genomics in their bodies. Now we have prepared the rebuttal, or we have prepared the critics and the rebuttal necessarily. Critical misuse of resources. Maybe the negative side could say there is critical misuse of resources in Africa. That's why the genomic and genomic process should not be applied in this sector. The rebuttal is, while the initial monitoring costs exist, genomic technologies are rapid and can be cheaper, according to the KIP of 2025 record. For instance, when samples have been pulled, as also seen during COVID-19 pandemic, Abdulhamid of 2020 has said this situation. Also, WASH reduces AMR downstream genetic monitoring is proactive. Critics number 2 is limited infrastructure LMIS. While this is true, genomic tools are becoming more accessible and portable. According to the KIP 2025 detail, many African countries have made remarkable progress in building national AMR surveillance systems. MBA and Martins 2025 have talking about this. Unfair to small farmers. This critic can be rebutted as, without the proper support, it should not be a problem. However, this view assumes punitive top-down approach, which is not necessary. Genetic monitoring can be inclusive with policy that empower small farmers, according to the Barrow records of 2021. Genomic surveillance generally of livestock for AMR is not a mere tool, but there is a proactive cornerstone of a true One Health strategy that can enable early detection, accurate modeling, and prediction of zoonotic pathogens risk, targeted interventions before resistant strains spill over into the human population. While concerns about cost, infrastructure, potential burdens of small farmers are valid, evidence from some African countries demonstrates that these challenges can be addressed through inclusive and supportive approach. By staffing genomic monitoring is livestock farming, we choose prevention over the reaction, safeguarding both animal and human health against the growing threat of zoonotic AMR in a way that no downstream measure alone can achieve. There are some references that we passed through. For example, we've passed through Chiguzi, VA and Cote of this transforming antibiotic resistance mitigations, Edro of 2023 contribution to prospects and trends of livestock production in sub-Saharan Africa, then Espinosa, FAO 2020, Gunasera, Fidro, Herman of 2023 health hazards, and so many. Abdulrahimid, Builder of Great of Swallen 2020, Al-Madok of 2023, the relevance of livestock husbandry in the context of food security. We have used also MBA, we have used Meriko Ibskeka of Chua, Panjimana, and of 2025, drug resistant zoonotic salmonella strains, and there are a lot of references. I could leave you to throw it and learn about it that have reach of knowledge and speaking about this situation of zoonotic transmission effects to the people of Africa. Thank you for listening. Feel free to ask any question from us. We will be quick responding you, and all in all, we just say thank you, goodbye. You have been with me, Selemani Sango from Dar es Salaam, Tanzania, and my friend Bashir Meriko from Malawi. As Group O, From group number 8, we thank you very much.